my dear little champions, I welcome you all to solve top 30 questions with me from the chapter Biotechnology, Principles and Processes, which is a very interesting chapter. And this particular video is for KCET 2025 aspirants, my dear children. So if you are someone who have opted for biology, then this video could save you in a lot of ways. Well, I have included all the previous year questions from this particular chapter, so you don't have to run around everywhere. And I'm telling you, I would be telling the answers in detail such that you understand the concepts as well. Works? Now there is a rule. If I am doing something, I have a rule. That is, whenever I show you a question, please pause the video and try to answer it by yourself. Now, the moment you realize that, okay, I'm not able to do, you have to turn it on and listen to me what I'm saying. And one more thing, participation is a must thing. So if you're not participating and telling me the answers over here, then I would be feeling really bad. So I don't want that. I want you to participate. Whether you say wrong answer, whether you say correct answer, everything is going to be really motivating me to solve more questions for you. Okay, and just your efforts matter, as I always say. So put down your efforts. Tell me, ma'am, this is the answer. I could correct you. I will tell you the correct way of solving things. But if you're not going to show that effort, then who will understand that who is trying or who is studying? So I want to know whether you are actually getting benefit of it, whether you are watching this video or not. Okay, so definitely participate. So let's start with the first question that we have over here out of the 30 questions. PCR is used for. What do you mean by PCR? PCR stands for polymerase chain reaction. Okay, now PCR is basically used for increasing the number of DNA. For example, I have a DNA fragment and I got it from a crime site. Now, I want to investigate on this particular DNA. I want to do so many things to find out who is the person behind the crime. So basically, this particular strand of DNA is not enough for me. I have to increase its number. So I have to copy this particular thing, just like how we make photocopies, right? Similarly, I want to increase its number. So this process in scientific terms is known as DNA amplification, where I am amplifying, where I am increasing the copy number of DNA. So PCR, polymerase chain reaction, is actually used for what? This particular process, which is what? Which is DNA amplification. DNA isolation means separation of the DNA. Okay, you want to isolate the DNA. DNA ligation means basically you want to join two DNA strands. DNA digestion means you want to break them down. So no, I'm not wanting all this thing from PCR. For PCR, basically it is for what? DNA amplification. Works? Chalo. Next question. Okay, which of these is not a method to make host cells competent to take up DNA? What do you mean by making host cells competent? For example, there is a cell and I want this particular cell to have the gene of interest. Okay, one gene of interest is there. I want to put it in there. So this method of basically transforming or basically putting my gene of interest into the host cell is what I mean by to make it competent. Okay, for example, now the host cell has the gene of interest. That means it is finally a competent one. Fine. Now, what all other methods which is not being used over here? So I say this and this is something that is being used. How can I explain? How can I explain that? Let's start with this. So biolistic, or what do you call it, gene gun. It is a method where basically a plant cell is there. It's a plant cell, okay? And what happens is that plant cell will have its own nucleus and everything, right? So I am basically using a technique which is known as gene gun, where I am using microparticles of tungsten or gold and I am surrounding it with the DNA of interest. I am putting my DNA of interest over this particular microparticle, and then with high pressure I am basically like gun, no, when you shoot. Similar to that, with high pressure, the tungsten or the gold, whatever thing is there, it gets inside the nucleus with the DNA wrapped around it. So basically I am inserting my gene of interest over here. So biolistics or gene gun is used for making the plant cell competent. Next thing, micro-injection. Micro-injection is where I am using an animal cell. Okay, and over here I would not prefer using gene gun. Why were we using gene gun over here? Because plant cell has an extra layer which is kind of thick. It is known as cell wall. But over here in animal cell, we do not have that. So basically, I can just use micro injection, where I am taking an injection sort of thing and I am putting my gene of interest with the help of the injection into the nucleus. This is known as micro injection. So again, I am making the host cell competent. These are the host cells where the gene of interest has to be put. Now, use of disarmed pathogen vectors. Now, this is another method. What do you mean by disarmed pathogen vector? For example, there was a bacteria, okay, which has its own plasmid and things like that, and this particular bacteria was not a good guy. Okay, was not a good guy. Why? Because it had some gene which could cause trouble when any organism is infected with it. For example, this particular bacteria is going to infect this particular plant cell. That means it is going to infect and cause little trouble. But now what I am doing is that the gene which could actually cause trouble, I am removing it. I am removing it. When I am removing it, I will say it is disarmed, as in does not have arms. No, it is what kind of safe. Okay, when I say there is a soldier with all the rifle and everything, I say he is armed. Now, when he does not have rifle and anything, things like that, I say he is disarmed. This nice time to attack, the terrorist could say that. Okay, so basically over here, similarly, if I am removing that gene that was causing the trouble, I will say that this particular thing, pathogen, okay, is what? Disarmed. Now, I would call it a vector. I would call the same disarmed pathogen as a vector when basically instead of this particular gene which was causing the trouble, I put my gene of interest, and since it is a pathogen, it will obviously go and infect, but this time without that gene which was causing the trouble. So, it will similarly go and infect the plant cell. It will go and infect the plant cell, and it will transfer the gene of interest over here. So, at this point of time, isn't it acting as a vector because it is transporting things from here to here? Vectors are things that transport things from A to point B. So, over here it is transporting things from here to the plant cell. So, anyways, I am putting at the end, what I am doing is that I am putting my gene of interest over here inside the plant cell. So, use of disarmed pathogen vectors is a correct way. Okay, can I put armed pathogen vectors? No. If it is armed pathogen vectors, it will cause trouble. When it causes infection, the plant cell will die. I don't want that. I removed that gene, I put my gene of interest instead, and then I put it. Okay. So, in this way also, basically, I am making the host competent to take up the DNA. So, it's all cool. Until now, it's all cool. But what do you mean by elution? Elution has nothing to do with anything over here. So, when I put DNA fragments in gel electrophoresis chamber and they get separated like this, no? After that, when I remove these pieces of DNA, this process is known as what, children? Elution. When I am separating this particular DNA strand from the gel, I call this process as what? Elution. It has nothing to do with making the cells or host cells competent to take up the DNA. So, elution is the wrong answer. This came in 2023, and elution is something that is given in your textbook. If you read the gel electrophoresis part, you will get to know the answer. Cool? Next question. Next question. Okay. Select the correct statements from the following. Let's see. DNA from one organism will not bind. I think it's bind. DNA from other organism. Okay. Genetic engineering works only on animals and not yet successfully used on plants. I don't think so that's true. It works, no? You have made BT cotton and things like that. There are no risk factors associated with our DNA technology. So, rDNA as in recombinant DNA technology. Do you think there is no risk factor? No, there are risk factors. Obviously, you are messing up with DNA. So, there are risk factors. So, this is again wrong statement. And DNA from one organism will not bind DNA from other organism. That is wrong. Okay, we have so much things in common, so it has the whole right to bind, so it's not a problem. Now we are supposed to find what, children? Correct statement. So you have to underline correct, because sometimes you might look for the wrong statement. So basically the correct statement will be the D1, because that is the only one left. The first step in PCR is heating, which is used to separate both the strands of gene of interest. So basically, if this is a DNA strand, we have to separate it out, and for that we need high temperature, or we need an enzyme that is known as DNA helicase. So over here in the case of PCR, polymerase chain reaction, with the help of high temperature and all, we are separating the two strands of DNA. Okay, then we will make a copy of it. So that is the first step, and that is known as denaturation. So this is a correct option, whereas others that you see over here, they're all wrong, my champions. Okay, works? Next question. From the following tools, techniques of genetic engineering, identify those which are required for cloning a bacterial gene. Okay, so we have bacterial gene, and we are putting where, children? In animal cell, and choose the correct option. So bacterial gene is there, and I am putting it in animal cell. So basically the first thing, over here that is written as endonuclease. Will I need endonuclease? Endonuclease is the enzyme that will cut the DNA. Obviously, I will need, because for example, this is the bacterial plasmid. I am cutting this particular gene. I want this particular gene. Obviously, I need enzyme to cut it, and that enzyme will be what? Restriction endonuclease. Endonuclease as in the one that cuts in the inside the DNA. If this is a DNA strand, I am cutting at the inside level. No, this is endonuclease. So definitely I will be needing this. Second is ligase. What does ligase means? For example, I took this particular DNA. Okay, I took this particular DNA, and now I am putting it inside the DNA of host. So basically this end and this end has to come together, and that happens with the help of ligase. So I need ligase also. Agrobacterium tumefaciens. This is a bacteria which is acting as a pathogen vector. Right? It is a disarmed pathogen vector which is being used always. Well, Agrobacterium tumefaciens is not used in animal cells. You are talking about animal cells, and hence you are not going to use Agrobacterium tumefaciens. It is used in plant cells. So this becomes something that we do not need. We are asked to find things that we need. Okay, tools that we need. Micro injection. So micro injection, do you think we need it in the case of animal cells? I told you that in the case of animal cells, you would be needing micro injection. Okay, micro injection is where you are inserting the gene of interest with the help of a thing like that, a tool. Okay, so that is micro injection that you use, and it is used in the case of animal cells. So this is something that we need. This. Okay. Now gene gun. Do we need gene gun? Gene gun is used in the case of plant cells since they have the cell wall. It is not used over here. So let's cut it off. Cellulase. Do you think we need cellulase by any chance? For example, bacterial gene we have to collect, but the bacteria is also covered by some peptidoglycan and things like that. Its cell wall is made up of peptidoglycan. So I don't need cellulase enzyme for that. I might need this enzyme, that is what? Lysozyme. I might need lysozyme for breaking open the cell wall of the bacteria. If it was a plant cell, children, if it is a plant cell, then I will be needing cellulase, because the plant cell wall is made up of cellulose. So basically what I am doing is I am taking the DNA from here, I am putting it some other organism. So anyways, I have to bypass the cell membrane also, whatever it is there. For example, this is a bacteria, it will have a cell wall made up of what? Peptidoglycan. And for this, breaking this down, you need an enzyme, that is lysozyme. This is an animal cell, it does not have cell wall, so I don't need any specific enzymes like that. Okay, suppose it was a plant cell, then I might have needed what? Cellulase also, because it will be needed for breaking down the cell wall of the plant cell if it was a plant cell. So basically I don't need this, I might need this. Now electrophoresis, do I need this, children? Electrophoresis is basically, I want to have a DNA strand, I want to separate it down on the basis of its size. So the smaller size one they separate faster, okay, and the larger size one they stay there. So I will take the smaller size one and I will put it over here. So that is the use of electrophoresis, and I need electrophoresis in general for anything. I would be needing it for anything, okay, for separation of the DNA fragments. So basically I am okay with having 1, 2, 4, 6 and 8, nothing else. Okay, these two options are kind of same. Wait. Okay, this is 7. Okay, now let's see. So things that we do not need is 3. If 3 is coming in any, that will be wrong. So you see 3 is coming here, so let's keep it wrong. 3 is coming over here, 3 is coming over here as well. What are we left with? We are left with 1, 2 and 4 and 8. Let's see, 1, 2, 4 and 8. Yes, we need all these things. So we say that this is the answer. Lysozyme was not included over here. It's fine. You have other options that are included. You can go for that. This came in 2023. So again, if you're not thorough with your NCERT, in NCERT all these things are mentioned. Now they have made a question out of so many concepts, right? So I would request you to go have a quick read of NCERT. And if you are not understanding the concept, basically I have made video for you in the case of NEET. Okay, so for NEET aspirants I made it, but it will be really useful for you as well. Okay, because certain questions are being trained from the NEET, the easier questions from NEET are being picked up for KCET also. I have noticed that. So you can go give a watch. Works? Cool? Chalo, let's go ahead. Gel electrophoresis is used for. Why do we think you need gel electrophoresis? I already told you what do you mean by gel electrophoresis. Over here I have a chamber, and this chamber will be having certain wells. Okay, in this well I will be putting my DNA. The DNA that I want to separate on the basis of what, children? The fragment length. Okay, if it is shorter, it will move faster. Where to move, where it is moving? So what I am doing is, I have an electrode over here, I have an electrode over here. This electrode, I will say it's cathode. Okay, it's the negative one, and this is the positive one, which I call as what? Anode. Now DNA is negatively charged. If you do not know it now. DNA is what, children? Negatively charged. So what happens? This also is negative, and the DNA which I am putting is also negative. So they will move, repel and will move towards the opposite pole, which is the anode. Okay, it will move towards the opposite pole, which is the anode. Now the DNA fragments which are smaller in size, they would be moving faster. Okay, and the one that are larger in size, they would be moving, what, children? Slower. So on the basis of this, you are separating the DNA fragments according to their what, children? You are separating them according to their size. Okay, please remember, anode is positive, anion is negative. Cation is positive, but cathode is negative. How can you remember? Cation, cations are positive. So cats have paws. You can remember like that. Cats have paws. So cations are positive. But cathode, on the other hand, it will be negative, anode will be positive. And you should be knowing about all this thing, because questions have been framed, you will see it. Okay. So gel electrophoresis is used for separation of DNA fragments according to the size. Is it correct? Yes. But let's read the other things. Construction of recombinant DNA by joining with cloning vectors? No. Cutting of DNA into fragments? No. Cutting of DNA molecule? No, children. It will be what? Separation of DNA according to its size. This came in 2022, and please remember this thing. It will be very, very important for solving other questions as well. I said that DNA fragments move faster, which are smaller. The smaller ones, they will move faster. I never said that it will move first. Okay, I never said that it will be the one to move first. I said it moves faster. So probably first you might have got the bigger DNA, then the smaller DNA might have moved. So please don't go put a correct statement when it put a correct option when it is said that DNA always the smaller fragments will be moving first. No, it will be moving faster. It can move faster after a point of time also, but it is not always the one that will move first. It can be any fragment that can move first. Okay, because it is negative, it will repel, no? So everything anyway have to move. Okay, an antibiotic resistant gene in vector usually helps in the selection of what? Antibiotic resistant gene, as in, for example, in the vectors that we choose. No, for example, I have a plasmid vector. Vector is something that will transport my gene of interest. Okay, so in this plasmid vector, which belongs to a bacteria, okay, I have an antibiotic resistant gene, that is, for example, ampicillin resistance, and I have another one which is what? Tetracycline resistant. Both of them are antibiotics. Okay, so basically, what do I mean by antibiotic resistant gene? When I am trying to grow this particular bacteria in a medium that has ampicillin, which is an antibiotic, it will still grow because it has the antibiotic resistant gene, ampicillin resistant gene. If I am growing it in tetracycline medium, still it will grow because it has tetracycline resistant gene. Now, when does this antibiotic resistant gene becomes useful and how? So basically, suppose this is tetracycline resistant gene. Okay, so initially, remember this particular thing, that this particular bacteria, whoever had this plasmid, whenever I am growing it in tetracycline resistant gene, it was able to grow. Okay, whenever I was growing it in ampicillin resistant gene, still it was growing. Now the situation is where I am putting my gene of interest over here. I am going to put my gene of interest over here in this particular site. So won't I lose the tetracycline resistant property? I would lose that. Now, basically, I am putting this particular bacteria with the new plasmid, having ampicillin resistance, and instead of tetracycline resistance, I have my gene of interest. Now, when I am trying to grow it in ampicillin resistant media, will it grow? When I am trying to grow it in ampicillin resistant media, ampicillin medium, will it grow? Yes, because it has still the resistant gene for ampicillin. But when I grow it in tetracycline 1, will it grow, children? Will this particular thing grow? It would not be growing. Why? Why will it not be growing? Because you have removed that resistant gene and instead you have put gene of interest. So basically, can I say now that this particular bacteria which is there, it has transformed Can I say it has transformed from this to this? Yes, it has transformed. Now, transformed as in basically, just a second, okay, wait. Transformed as in when I am putting this particular bacteria into some host, okay, when I am putting this bacteria into some host and this has this particular plasmid, okay, just a second, okay, I will say that the host cell has this particular bacteria, or particularly I will say that they have the gene of interest when I would understand the fact that it is not growing in tetracycline medium. So I will say that this particular host cell is now what? It is transformed. Suppose there was another host cell where I was putting the normal one over here, okay, or basically, to not make you confused, let's suppose there is a normal host cell over here. There is a normal host cell over here, and basically this host cell is not able to survive in ampicillin and both tetracycline. I would say that it is not transformed. It is not transformed. If it was transformed, it would have received this particular bacteria. It is not transformed, since because of that only I am saying that it is not resistant to ampicillin nor it is resistant to tetracycline. Okay, I hope you got it. If you didn't get it, I'll tell once more. Listen, this is a normal bacteria which has ampicillin and tetracycline resistant gene. Now I want to put this particular bacteria, or basically I want to bring some changes and then I want to put it inside a host. Suppose this yellow one that you see over here is the host. Okay, so I have host cell, I have bacteria, I want them to infect. Some of the host cell might get infected, some of the host cell might not get infected. Okay, the one that get infected, I'll say that they are transformed. The one that does not, I'll say that they are not transformed. So basically, how will I get to know whether they are transformed or not? The transformed one, they will show resistance to what? Some antibiotic. For example, over here the transformed one is showing what? Resistance to what? Ampicillin. It will still grow in ampicillin because it has the ampicillin resistant gene. Over here, if I try to grow in ampicillin resistant gene, still it will not grow because it does not have the bacteria which has the gene for ampicillin resistant. Neither it will go for tetracycline also. So I will say this is non-transformed, whereas this one, this particular host cell is transformed. This host is now transformed to have the gene of interest. Works, or probably it is transformed to have this infection by this particular bacteria. That will be more correct. So I will say with the help of antibiotic resistant gene, what I am doing, I am helping in finding the transformed cell. Okay, it is helping me in finding the transformed cell. Now there is one more situation where there is a host cell and there is a bacterial cell. In this bacterial cell, I will say it has both antibiotic resistance, ampicillin and what? Tetracycline. Still I will say it is transformed. Both of them, they are transformed because both of them are infected by bacteria, which is why they are resistant to both ampicillin. This one is resistant to tetracycline also, that means it is also transformed, but it is transformed, but I am not sure whether it has the gene of interest. To make sure of that, what will I do? I will try to grow it in tetracycline medium. Only the ones that does not show tetracycline resistance will be the one that will be competent also. Okay, or basically that will be the one recombinant as well. So I mean to say transformed is different from recombinants. Recombinants is the one where I have the new combination of things. Okay, over here I have the one that had new combination. This is the recombinant one. Okay, I have my gene of interest. Over here this is transformed. Just like this particular host cell is also transformed because it has the bacterial cell over here. But over here when I say it does not have gene of interest, which is why it is able to grow in both ampicillin and tetracycline. Over here it will be able to grow just in ampicillin but not tetracycline. So basically first thing that I would be doing with the antibiotic resistant gene is to find whether it is transformed or not. Once I know that it is transformed, I will check whether it is recombinant or not. That is the second option. So whether it is competent cell or not, that will be the second option. So competent cell, recombinant cell is the same thing. So I would get to know later on. So the first thing will be always transformed. So you get confused between this and this, which is why I explained that also. Okay. Works? Next, the term molecular scissors refers to molecular scissors as in molecular. In molecular level, if something is cutting something, you call them molecular scissors. In the case of biotechnology, there is only one molecular scissor which you call as restriction enzymes. So restriction enzymes are the one that will be cutting the DNA strands, okay, which is why you call them molecular scissors. This came in 2022. Works? The other question took some time for me to explain because it is an important thing. There are so many questions around it, so please don't judge. Okay. Next question. ROP gene, which codes for the protein involved in the replication of the plasmid PBR322 in E. coli, is located at restriction site of. So basically, I have a plasmid, and this particular plasmid has the certain sites. I told you, right? Ampicillin resistant. There is what? Tetracycline resistant. In this resistant gene only, there might be site for enzymes to cut also. Okay, for example, you have BAMH1 over here, SAL1 over here. So that means at this particular site, restriction enzyme BAMH1 can make a cut. That is what you mean. So similarly, you have a gene like this which is known as ROP gene. What is it? ROP gene, which is very important for replication. Okay, for increasing the number of the DNA, for increasing the copy number of DNA. So basically, in the ROP gene only, there is a site for restriction enzyme. There is a site for one restriction enzyme, and what is that restriction enzyme is the question. Okay, this plasmid that you see is known as PBR322 in E. coli. This is found in E. coli, this particular plasmid. Now basically, this site is owned by an enzyme which is known as PUV2. This is what? PUV2. Please remember, in ROP gene you just have one, and that is PUV2. This is 2021 question. So you should be knowing the site for each and everything. For example, over here in the ampicillin resistant gene, you have two. One is PVU1, one is the other PVU1, and one is the other one. Okay, here BAMH1 and SAL1. Over here you have EcoR1, you have HIN3, and a lot of things. So basically you should be knowing the sites for each of them. Okay, especially when it is overlapping. So this gene is for ROP, but this is also a site for PUV2. So you should be knowing all this thing. Go see the picture of that plasmid. Very important. Next, what does the sample of given base sequence represent? So you have GAATTC in 5 to 3' version. Again over here, this is 5'. Okay, you have GAATTC. So in both the side, when you read it, is the same thing. 5 GAATTC, 5 GAATTC. These sites are known as palindromic sequence. These sequences are known as what, children? Palindromic sequence. There is nothing else. Palindromic sequence as in when you read from both the direction, if it's same thing. For example, you have Malayalam. From here to here, if you read Malayalam, it's Malayalam only. Here to here also, it is Malayalam. So what do you call the sequence as? Palindromic sequences. And what is the use of this? This is used in the case of cutting down things. This is used by restriction enzymes. So restriction enzymes have certain sequences, certain sites where they cut. So this is very useful in their case. Okay. Cool. 2021. So whenever you see both the things are read in the same way, in same polarity, 5 to 3, 5 to 3 is the same thing, then they are palindromic sequence. There are so many questions associated with it. I will be showing you that as well. Now the 10th question. From the given combinational steps in PCR, identify the enzyme dependent step. So there are 3 steps. One is denaturation, where you have your DNA and you are separating the strands altogether. This is known as denaturation. Second one is known as annealing. Second one is known as annealing, where basically you are putting primer on both the ends. You are putting a primer on both the ends. So suppose this is 3 dash, this is 5 dash, and this is 5 dash, this is 3 dash. You are putting primer at 3 dash end. Okay. Now why do you need to put the primer? Because for the next step you need one sequence over here for that. Okay. So basically, what do you call this step when you are putting the primer? You call this as annealing. And for the next step, which is known as what? Extension. The next step is known as extension. This is the first, this is the second, and the third. In extension, you need an enzyme which is known as DNA polymerase. What do you call it? DNA polymerase. In the case of PCR, you call the same enzyme as Taq polymerase, because it is taken from Thermus aquaticus. This particular polymerase, this particular enzyme is taken from Thermus aquaticus, which is a bacteria. So you call it Taq as in Thermus aquaticus. Taq polymerase. So this particular enzyme will be later extending the DNA strand. So for doing that, it needs a primer, which is why you did annealing first. Okay, so this is a step where you would be needing what, children? You would be needing enzyme. So the answer for this is, from the given combination of steps in PCR, identify the enzyme dependent step. It is extension only. For extension, you would be needing an enzyme, that is Taq polymerase or DNA polymerase. Okay, why do you need a bacterial enzyme over here? Because it is the one that will be able to survive in high heat. So basically you are doing in 70 degrees Celsius, 95 degrees Celsius. The first step is done in 95 degrees Celsius. 95, yeah, almost around that. And annealing is done in 50 degrees Celsius. Extension is done in 70 degrees Celsius. So you have high temperature. So you should be having enzymes that can survive in high temperature, right? So which is why you use Taq polymerase, which is why I said extension is a step where you will be needing enzyme. Cool? This came in 2020. Next question. Biolistics method is suitable for gene transfer into. Biolistics means what? Gene gun. And gene gun will be used when the cell wall is kind of very strong. And the strong cell wall is found in what? Plant cells, because their cell wall is made up of what, children? Their cell wall is made up of cellulose. So which is why you are using biolistics, or the same thing. What do you mean by this? This is actually the same thing, that is gene gun, where what you are doing is that you are taking microparticles of tungsten or gold, you are covering it with the DNA of interest and you are putting it in the host body. Okay, the plant cells body. Fine, works. For animal cells, what do you use? For animal cells, what do you use, children? You have this micro injection. For bacteria, you have heat shock treatment. Okay, where the bacterial cell is made to have a cold first, then they put in ice first, then they are given heat. In this way, the temperature and everything changes, it creates pores and things can later enter into it. Okay, works. Next thing, identify the labels M and S in the following agarose gel electrophoresis representation. So over here, this is the well, and you see things are moving from here to here. This is the well, obviously things will be moving from here to here. And there is S strand, there is M strand. M strand, as I already told you, the larger size ones will stay back, the smaller size ones will be moving faster. So over here you will be finding the smaller size one. So M, can I say it's small DNA bands? No. Can I say M is digested DNA bands and S is undigested DNA bands? No, children, all are digested. They are all cut and they are all going through the gel electrophoresis chamber. So this is also wrong. M is hybridized DNA bands. Hybridized as in? No, it does not make any sense. They are not hybridized or things like that. So basically I will say this is wrong. Now D says M is the largest DNA band and S is the smallest DNA bands. This is actually correct. This would be the smaller one and this would be the larger ones. So this becomes a correct option. 2020 question. I told you, right? This particular topic gives you a lot of questions. So you have to be very much aware of the concepts. Cool? Next, suppose DNA samples collected for DNA fingerprinting analysis are less. Okay, suppose I took from some DNA for DNA fingerprinting analysis, as in I want to find out whose DNA is this particular thing. Okay, I want to match things, lot of things. Which of the following techniques is helpful to make the sample sufficient for above analysis? And I have too less amount of DNA, I want to increase its number. So which one do you think would be very helpful? DNA probing, electrophoresis. Electrophoresis basically means you are separating things. Chromatography, which is a whole different technique. PCR, which step will I use? When I have small DNA fragment, I want to increase its number. Okay, at this particular point of time, I would prefer DNA amplification, and DNA amplification is done by what? PCR, polymerase chain reaction. This is a direct question, came in 2020, very easy, right? So we can give it as an easy tag and do this. It's easy things. Next, in cloning vectors, antibiotic resistant genes are helpful for. So we did the same question which took a lot of time, which is why I said this will help you later on. So in cloning vectors, okay, cloning vectors as in the ones that can create its clone. For example, I have a vector and over here I am putting my gene of interest and then I am allowing it to clone, increase its number. So basically these are known as cloning vectors. In a way, when they are making their own clone, they are taking my gene of interest also, making clone for it also. So I am increasing my number anyway. Anyways, over here in the case of these vectors, antibiotic resistant genes are helpful for. Why do you need antibiotic resistant genes? Transfer of foreign gene into the host, selection of recombinants, making the host cells competent, cleaving of vector by REN. REN as in restriction endonuclease. I would be using for selection of recombinants. As I said, for example, I have an antibiotic resistant gene over here and one over here. I am cutting this gene and putting this gene of interest. Okay, then I am trying to grow it in a medium that has tetracycline, it won't be able to grow. That means this particular thing is what now? A recombinant. So basically I am able to select the recombinants or the one that has new combination with the help of the antibiotic resistant gene. So this would be the correct answer. Cool? Next question. A student while extracting DNA from Aspergillus fungus, enzyme to break open the cell wall would be. So Aspergillus is a fungus. Now if this is a fungus, I want to take the gene from the fungus, I want to break the cell wall, I would have to break the cell wall, and in the case of fungus, the cell wall is made up of something that is known as what, children? Chitin. So what enzyme will be needed to break down chitin? I will be needing chitinase. So once I break it down with the help of chitinase, I can get into it and take the gene of interest. Okay, or probably extract the DNA. So you should be knowing plant cell always have cellulose, animal cell has what? Plant cell has cell wall made up of cellulose, which is why they need cellulase enzyme. Bacterial cell have their cell wall made up of peptidoglycan, which is why we say they need lysozyme enzyme, which is something that can break the bacterial cell wall. And chitin is something that makes up cell wall of fungi, that's why you need chitinase. Pectin is something else, pectinase, so do not go for that. It is enough, so just remember that works. These three enzymes, if you remember, it will be more than enough. Okay. Next question. Identify the DNA sequence which can be cut using EcoR1. EcoR1 is a restriction enzyme, and each restriction enzyme have their own recognition site, as in they have a sequence in which they will work. They will check for that sequence, they will cut in that particular sequence. I told you, right, that particular BAM-H1 was having gene in the tetracycline 1. So basically they have their own site. This is very specific. So for EcoR1, the site is GAATTC. This is in 5' to 3' direction. This is the sequence for EcoR1. This is given in your NCERT and you have to remember it. GAATTC, 5' to 3' direction. Similarly, opposite if you make, it will be GAATTC. I told you, TTC. This is again 5' to 3' direction. These are the two strands. So they will be making a cut over here and making a cut over here whenever they see the site. Okay? Please remember it. They have the sites which are palindrome to each other. So this is a site, let's search for it. So DNA sequence over here, do I find GAATTC? But it's in 3' to 5' direction. I want 5' to 3'. So let's see. Over here 5' to 3' is there, GAATTC. Okay, I got it. But here also have to find it like that. 5' GAATTC. So this is correct. But let's see other options. Over here I have GAATTC, but this is 3' to 5'. That's a problem. I want in 5. On the 5 side there will be G. So this is again wrong. I cannot include this. Let's see this. 3' GAATTC. This is again 3' to 5'. This is again wrong. So that's why I say you have to know the polarity as well. So 5' to 3' it will be GAATTC. So this becomes your answer. This is the restriction site for EcoR1. This is given in your NCERT. You have to remember it. And for HIND also, for HIND 2 enzyme, I suppose, they have not given the site, but they have told there are 6 bases. For example, over here 1, 2, 3, 4, 5, 6. Here also you have 6. Similarly, in the case of HIND2 also they have 6 bases. And that's the first enzyme that was restriction enzyme that was found out. So basically you have to remember that at least they had 6. Okay, and the name also. So let's go ahead. Identify the palindromic sequence in the following base sequences. Listen how many times the questions have come, you see. Now palindromic, as in it should be same from both the sides. So C, G, A, if you see C is here, but here there is no C, I cut this off. If you see C is there, G is there, I cut this off. G is there, G is there. Let's see. G, G, A, T, C, C. G, G, A, T, C, C. So this is correct, but let's see the other option. G is here, C is here. This is again wrong, because if G is here, G has to be here also, because it should palindromic, right? So it has to be the same thing. So this becomes your correct answer. This is 2018 question, my champions. Works? So I hope you're getting to know or understand what do you mean by palindromic sequences. Next thing, question number 18. The technique of bombarding plant cells with high velocity microparticles of gold or tungsten coated with DNA is known as microinjection, biolistic method, heat shock method, by disarmed pathogen vector. What will be the answer for this, children? It will be biolistic method. Biolistic as in gene gun, and this is what you're talking about over here. I told you, you are using microparticles of gold and tungsten, you are coating with DNA and then you are inserting it with high velocity. This is known as gene gun or ballistic method. This is used just in the case of plants. 2018 question. Okay, next question. Choose the bacterium which is not a source of REN. REN as in restriction endonuclease, or the enzyme, okay, which is not the source of restriction endonuclease. So from Haemophilus influenzae you get, so that is why Hind, I told you Hind 3, Hind 2. So this comes from Haemophilus influenzae only. Escherichia coli also gives me enzyme. I saw it right now. It is EcoR1. Okay, this is got from Escherichia coli. Bacillus amylocufaciens, it also gives you enzyme. Agrobacterium tumefaciens, this is not something that can give you enzyme. Okay, Agrobacterium tumefaciens is the one which always acts as a pathogen vector. It's the one, okay, it is actually a pathogen, a bacteria that causes trouble or infection in the case of plant cells. Okay, so what did they do? No, the biotechnology people, they, for example, this is the Agrobacterium tumefaciens. It had its own plasmid with the gene that was causing the trouble. You call it the TI, tumor inducing gene. Okay, so over here what they did was, okay, they changed this particular, they removed this particular part and they put the gene of interest. Now this poor bacteria does not know that it cannot cause any trouble. It still goes and infects the plant, but when it is going and infecting the plant, it is not able to transfer that particular DNA strand which is causing the trouble. Instead they put the gene of interest. So this is a disarmed pathogen vector. It does not have, it is not a source of restriction endonuclease. All of the others are. So basically this is the answer. Okay, and this always acts as disarmed pathogen vector in the case of plants exclusively. Cool. 20th question. Restriction endonucleases are isolated from some bacteria. Their role in bacteria is to. So why do you think that I said that these guys that you see over here, they all have restriction endonucleases. There is a source of restriction endonucleases. Why do you think they have restriction endonucleases? Why do you think they have the enzyme? So what happens is that, for example, this is a bacteria, okay, and this is their DNA. This is their DNA. And suppose there is one virus that came over here and tried to infect it. This virus gives its genetic material inside, and slowly what it does is that it puts a genetic material over here in the bacteria's body and will make copies of it. Okay, use bacteria's machinery to make copy of it. Now what do they actually do is, bacteria, they instead release, basically they have certain enzymes known as restriction enzymes. These enzymes will go and cleave this viral DNA. They will go and digest this viral DNA. They will go and cut this. So basically, don't you think this is acting as a defense against viruses? They have their own restriction enzymes because it will be acting as a defense against viruses, such that if I'm a bacteria, I'm cutting the genetic material off, this cannot come in and get incorporated over here. Get it? So what becomes the answer? This becomes the answer. That is, it acts as a defense against virus. This is question number 20. Works? Okay. Next question. From which bacterium the REN SAL is isolated? SAL is written, no? SAL. The first letter of an enzyme comes from the genus name. So over here it can be this or it can be this. Okay, the second two letters come from the species name. So over here, if I find AL over here, that will be correct. So this is not the one. This is AU. Over here I see AL. That means this is the one that is correct. So it is Streptococcus albus, from where I got the restriction endonuclease, that is SAL. So please remember, first one is the first letter of genus name or generic name. Okay, over here AL are the two letters from the species name. Got it? Next question. A transformed bacterium with human gene fails to produce desired protein. The reason could be, okay, a transformed bacterium with human gene fails to produce desired proteins. How? Why? So for example, it's saying that this particular bacteria has our gene of interest over here. And we suppose that it will do what? Basically, the first step is DNA is made into mRNA. This mRNA is helpful in making what protein? This is known as transcription. This is known as translation. Now it can happen in the case of bacteria also, but the problem is that basically I put my gene of interest and now it is not making the protein. And what could have been the problem? That is the question. Why is it not making the desired protein? Usually it does. The problem could be that basically when the DNA makes the mRNA, okay, when we copy a DNA, no, our DNA or basically mRNA which is being formed over here, there is something else which basically has not the mRNA before it. Okay, wait. So this DNA makes the mRNA. This is a general thing that we say, but it does not directly make an mRNA. It forms something that is known as hnRNA. This hnRNA later undergoes some processing, okay, and then it forms mRNA. What is this processing? Basically, there are certain regions in our DNA which are the coding ones. You call them exons. What do you call them? You call them exons. So in between the exons, you have certain non-coding regions which are known as introns. But bacteria does not have that. Bacteria does not have that usually. So they do not have the enzymes which would cut this introns off and join the exons. But in our case, we have the enzymes which would cut this off and join to make a straight strand of exons. And you call this as the mRNA which is formed. So initially you have hnRNA, and this hnRNA undergoes certain processing to form mRNA which looks like this, and then later it forms protein. But now suppose I have put a DNA strand or probably something that has the introns within it already. Usually I choose something that does not have introns within it. Okay, but this time I didn't look for that. I put the introns also. Now in the bacteria there is no enzyme that could cut off this introns, right? Because they do not have that. So which is why the proteins are not being made. So human gene may have intron which bacteria cannot process. This could be the reason why they are not making up the protein. Always remember, bacteria does not have intron, so they do not have the machinery to cut this introns off to make a protein, to make a exon, to make a strand of exon like this, which is actually known as mRNA. So we have to make sure the gene of interest that we are putting does not have intron non-coding sites in them. Then only things would be working. Okay. So amino acids code for codon for human and bacteria differ? No children, they are same. Human protein is formed but degraded by bacteria? No children. The bacterial promoter gene cannot induce transcription of human gene? No, that is also not the reason. It's because the human gene may have intron which the bacteria cannot process. That is the thing. So this becomes the answer. Works? Next question. Most suitable method of introducing alien DNA into a plant cell. How many times this question have come? Into a plant cell, if you want to put alien DNA as in the new DNA, gene of interest, it will be biolistics, or basically what? The gene gun method. Again and again and again and again. So many times. So this is an important topic. Please remember that. 2016 question, and biolistics people usually forget because what they remember is what? Gene gun. So biolistic and gene gun is the same thing. Okay. Next question. Which one of the following statement is wrong with respect to separation of DNA fragments on gel electrophoresis? Okay. What is wrong? You have to find what is wrong. So underline what is wrong. Okay. The DNA fragments move towards anode under electric field through the matrix. Yes. The DNA fragments generally will be moving towards anode. I told you anions are negative, but anode is what? Positive. And DNA is negative, so DNA will be moving from cathode to anode. And it is moving through the matrix. It is also correct. This is correct. The commonly used matrix is agarose gel. Yes, the matrix that is used is what? Agarose gel. The DNA fragments resolve according to their size. So they move according to the size. That is also true. The smaller DNA fragments separate first. That is not true. I already told you. The smaller DNA fragments, they move faster. They always move faster. But it can be the bigger ones that separate first, and then later on the smaller fragments move faster. That can be the case. So it is not always true that they are the ones that separate first. That is a wrong thing to say. Okay. So it's a confusing thing, but I have solved it for you. I hope you will remember it. Cool. And always remember one more thing, there can be options which have come in NEET where they have said that DNA fragments move above the surface of agarose gel. No, the DNA fragments basically move within the agarose gel. Okay, it is moving through the agarose gel or through the matrix. Always remember that it's not on the surface. If it is written on the surface, if here the statement was DNA fragments move towards anode under the electric field on the surface of the matrix, then it would have been wrong. Okay, so it is always within the matrix it moves. Cool. And it is given in your NCERT textbook, where they have said that the gel which is used has sieving effect. Sieving effect as in it will be forming pores and that such that the DNA could be moving around. Cool. Okay. Which one of the following statement is not correct about a plasmid? Plasmid is, for example, this is a bacteria. Bacteria will have its own, what say, chromosomal DNA, the DNA that will be coding for proteins and stuff like that. Apart from that, they would be having certain plasmids, which are double-stranded DNA only over here, which generally do not code for any proteins. Okay, but they have their origin of replication. They will keep on replicating itself and things like that. So they are just there. They are extra chromosomal DNA, which you call as plasmid. Okay, and this is being used in what say recombinant DNA technology. You usually cause changes over here. If you cause changes over here, this particular bacteria will be something else. We do not want that because it is coding for proteins, and that proteins basically define the bacteria. So we do not want to bring any changes over here. We use the plasmids for that. So which one of the following statement is not correct? We have to find that. It is circular DNA. As you can see over here, it is circular DNA. So this is true. It has its own origin of replication. Yes, it will have its own origin of replication. Origin of replication is a sequence which will allow the DNA polymerase and everything to come bind over there and then basically replicate. So origin of replication is something that you need. So yes, they have their origin of replication, which is why they replicate also. So that is true. It has the ability of autonomous replication. It has the ability of autonomous replication. Autonomous as in by itself only it can replicate. Yes, it can definitely do that. Its DNA is long as chromosomal DNA. The chromosomal DNA and this guy is totally different. It will be very short. Okay, plasmids are shorter. So I would say this is wrong statement. We had to find the non-correct or the wrong statement. So this is the wrong statement. Its DNA is not as long as the chromosomal DNA. Please remember that. So here it is written it's long as chromosomal DNA. It's a wrong statement. Cool. 2016 question. Next, illusion means what? Illusion means, I already told you, in a gel electrophoresis chamber, at the end when you have the DNA fragments being separated, no? Now we are cutting this DNA fragment off from the agarose gel. This process is known as elution. So, separation of DNA fragments on agarose gel is not elution. Cutting and extraction of DNA bands from the agarose gel is elution. You are cutting it off and you are extracting the DNA, that is known as elution. Making the DNA bands visible under UV radiation, no. Isolating alien DNA from the choice organism, no. This is the answer. Cutting and extracting of DNA bands from the agarose gel is what you mean by elution given in your NCERT. So with this you know that bioelastic and electrophoresis and palindromic sequences are something that is being usually asked. So you should be knowing very well about it. Okay. Next thing, with respect to DNA fragmentation, okay, which statements are correct? Statement A, gel electrophoresis and elution are two important processes. Yes, gel electrophoresis helps me in separating the DNA fragments, and elution helps me in extracting those fragments. That is important. Statement B, after staining with ethidium bromide, it has to be exposed to UV light. So basically, the problem is basically I have separated the DNA. The DNA won't be visible. Okay, where are the strands? It won't be visible in the gel electrophoresis chamber. So what do I do is, basically the strands which I have separated in gel electrophoresis, with the help of another sheet which is known as nitrocellulose sheet, I just put it like this and I transfer the whole set of DNA on this particular sheet. Now, suppose this is a sheet. Still the DNA is not visible over here. It won't be visible. So what do I have to do is, I have to treat this with ethidium bromide. Ethidium bromide to make it visible. Oh, this strand is over here. I have to understand that. For that, I have to treat this particular strands with ethidium bromide. But still it won't be visible until I put UV light. When I put UV light, what happens is that ethidium bromide will be giving a bright orange color, which is how I understand that, okay, the DNA fragments are over here. So after staining with ethidium bromide, it has to be exposed to UV light. Without exposing to UV light, you are messing things up. So statement A and statement B both are correct. And later I put this DNA, I take this DNA and use it for my use. So with respect to DNA fragmentation, if I say statement A and statement B, both of them are equally correct. Please remember, ethidium bromide gives bright orange color and it will be only visible if UV radiations are there. This is an important NEET question as well. Okay, so both the statements are correct. Both A and B are the correct statements. This came in 2015 My Champions. Works? Okay, next, which vector can clone a small fragment of DNA? So these things that you see over here, plasmid, bacterial artificial chromosome, yeast artificial chromosome, cosmid, all of them, they are going to act as vectors. I told you vectors are the things that will transport my gene of interest from one organism to other organism. Okay. So for example, mosquito is a vector. Why do I say mosquito is a vector? It has the parasite, it is transferring that parasite to me in the case of malaria. So that is acting as a vector, it is acting as a carrier. So over here, all these things that you see, they are acting as carrier. Plasmid, bacterial artificial chromosome is the one which is, okay, it is known as BAC and this is known as YAC. This is for, it transports things into bacteria. Okay, and this help in transporting things into what? Yeast, and then later yeast can be used or bacteria can be used for our use. This is artificially made. Okay, this was made in the lab. This is not something naturally found. This is made in the lab, and both of them, they carry larger fragments of DNA. Larger gene of interest or larger fragments of DNA. Both BAC and YAC. But when you talk about plasmid, they are the one that carries what? Smaller fragments. After plasmid comes cosmid. After cosmid comes these two guys. Okay, so in the order, this is the one that carries the smallest fragments, then comes cosmid, then comes these two. Okay, cool, please remember it. Next question, EcoR1 is what? Is a restriction enzyme. Okay, it is what? A restriction enzyme derived from that is Escherichia coli. So Escherichia coli, and R1 is the strain's name. Okay, it is the name of the strain. So used to join two DNA fragments, no children, it is DNA ligase which is used to that. It is a restriction enzyme, exactly correct. The abbreviation for bacterium Escherichia coli, no, it is not the abbreviation for the bacterium, it is the abbreviation for the enzyme. So EcoR1 is there. Okay, so basically the abbreviation for the bacteria will be E. coli. Over here we are basically talking about the enzymes, and over here it said it is plasmid. No, it is an enzyme, it's a restriction enzyme, and this question came in 2014. My champions. Works? The last question that we have over here, identify the DNA segment which is not a palindromic sequence. So it should not be a palindromic sequence, it should not be read in the same format from both the sides. Okay, so let's see. Over here we have G and we have C. So this is not a palindromic sequence. If it is a palindromic sequence, then if it's G, G, A, T, C, C over here, it should have also been G, G, A, T, C, C. It should have been like that, but it is not. But let's see the other things. Over here I have G, over here I have G. Let's see the things. G, A, A, T, T, C, G, A, A, T, T, C. This is the palindromic sequence. Over here, G, C, G, G, C, G. Okay, then G, C, C, G, C. Then G, C, C, G. Okay. Okay, but still it has sequence until this as same thing. So this is also palindromic sequence. So people might get confused over here. There's an extra G, ma'am. But okay, you can take it from here to here. That's fine. This is a sequence, palindromic sequence. It is read like that. Here also it is read like that from here. Okay, until this spot. Here also it can be taken until this spot. Then it's fine, right? Okay. Let's see this. C, C, G, G, G. Over here I have C, 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 then 2G. Okay, I can take it until here. Here also I can take it until here. There is one extra which is not there. Fine, I can. Anyways, if you see from here to here, isn't the same thing? If it's the same thing, then it has to be palindrome. So both, or basically three of them, if you see, they are palindromic sequences, but over here it is not a palindromic sequence, and you had to identify that only. So I hope you are able to understand the palindromic sequence. So as I said, with these many questions that we solved, I am able to understand one thing clearly. Palindromic sequence, restriction enzyme, biolistics, or basically the enzymes used for infecting plant cell and things like that, you should be knowing about that really well when you're doing this, and gel electrophoresis. All these topics are really important when you are, revising this particular chapter. Please keep notice of that and give ample amount of, study or time for understanding these concepts that I just told you right now. Okay, so this is how you will understand things. If you are not solving questions, you would not be understanding. So please try to solve as many questions as you can. From the NEET topics also you can, there is no problem. If you have time for doing so, then definitely do it. Don't think that, okay, I am doing NEET questions and I might get demotivated if I do not know. It's fine, just try it out, it will be helpful. You never know what type of questions could be framed. Okay, you saw a question which had everything together, all the concepts together. So I would recommend you should try it out. It is very, very easy. If you try it out, it will be good for you only. Okay, and if you have any doubts, my children, you can put it down in the comment section. I would be ready to solve all kinds of doubts that you are having over here related to the topic that I just taught. Now, if you want certain other chapters that I should be doing, you can tell me that also. Works for me, it should work for you as well. So that's all. If you think that this particular video was helpful, then definitely do not forget to subscribe to our channel, Deeksha Karnataka. And if there is someone who have got more than 10 questions to be correct, definitely tell me in the comment section. I would be so happy to know that. More than 10 and more than 15, you can definitely tell me in the comment section. And if you're someone who needs help, then also definitely let me know. Okay, so bye bye, take care, see you on the next day.